For close to 2,000 years, one of the most contested questions in the study of any human population has surrounded the Jewish people. Scattered by conquest and exile across Europe, North Africa, the Middle East, and parts of Asia, and separated from one another by vast distances, different languages, and centuries of almost no direct contact. Jewish communities in places as far apart as Poland, Yemen, Morocco, Iraq, and India nonetheless continue to describe themselves as one people, united by a shared ancestral link back to the ancient Israelites of the biblical Levant. Written into religious texts, preserved in oral tradition, and reinforced by shared prayers, calendars, and legal customs, practiced almost identically across communities that had never met one another. This claim of common descent is not a fringe belief. It has shaped Jewish identity, Jewish law, and Jewish political thought for more than 2 millennia. A rival scholarly argument has run directly against it for well over a century. Beginning with early 20th century historians and gaining renewed traction in more recent academic and popular writing, this counterargument holds that Jewish communities are not a genetically continuous people at all, but rather a loose collection of unrelated local populations who, at various points across history, adopted the Jewish religion through conversion, and who therefore share essentially no deeper biological connection to one another or to the ancient Israelites beyond a shared set of religious texts and practices. A closely related and even more specific version of this argument, known as the Khazar hypothesis, claims that the largest single Jewish community in the world today, the Ashkenazi Jews of Central and Eastern Europe, descend not from the Middle East at all, but primarily from the Khazars, a Turkic-speaking steppe empire based north of the Caucasus that is historically recorded to have adopted Judaism as its ruling religion around the 8th century, before collapsing several centuries later. 2 sharply opposed claims, one insisting on a single continuous bloodline running back thousands of years to ancient Israel, the other insisting on no meaningful shared ancestry at all, with a specific and historically dramatic alternative origin proposed for the largest Jewish community on earth. For most of the 20th century, this argument played out almost entirely through history, linguistics, and religious scholarship, with limited ability to test either claim directly. That has now changed. Over the past 2 decades, population geneticists have conducted a series of large-scale genome-wide studies specifically designed to test these competing claims directly, comparing autosomal DNA, Y chromosome lineages, and mitochondrial DNA across Ashkenazi, Sephardic, Mizrahi, North African, Yemeni, Ethiopian, and Indian Jewish communities, and setting the results against reference populations from across Europe, the Middle East, the Caucasus, and North Africa. The results didn't simply confirm one side of this argument and dismiss the other outright. They revealed something considerably more specific, considerably more historically layered, and centered on one particular ancient genetic connection that turned out to be more consistently and unmistakably preserved across these scattered, unrelated communities than almost anyone studying the question expected going in. To understand why this question proved so difficult to settle through history alone, it helps to understand the sheer scale of the Jewish diaspora and the enormous span of time and geography involved. Following the destruction of the Second Temple in the 1st century and a series of subsequent revolts against Roman rule, Jewish communities scattered outward from the Levant across the Mediterranean world, eventually establishing settlements stretching from Spain and Morocco in the west to Persia, Yemen, and India in the east, and from North Africa in the south to the emerging kingdoms of medieval Europe in the north. Each of these communities lived for centuries, in some cases well over 1,000 years, alongside dramatically different host populations, adopted different local languages, and in many cases showed clear evidence of at least some intermarriage and conversion, involving people from the surrounding non-Jewish population. Given that scale of separation, a claim of zero remaining biological connection between, for example, Ashkenazi Jews of Poland and Yemenite Jews of the Arabian Peninsula carries genuine surface plausibility, since these 2 communities had no meaningful contact with one another for well over 1,000 years, spoke entirely different languages, and lived within entirely different genetic neighborhoods. At the same time, the historically documented pattern of Jewish communities maintaining strict religious endogamy, marrying almost exclusively within their own community generation after generation, even while living as small minorities embedded within much larger host populations, gives the traditional claim of preserved shared ancestry its own genuine plausibility as well. Both claims, in other words, rested on real and defensible historical logic. What neither side had, until relatively recently, was the genomic technology capable of actually measuring the answer. This academic disagreement was never purely a matter of dry historical debate confined to university seminar rooms. The question of Jewish genetic continuity has carried real political and cultural weight for well over a century, invoked at different times by very different groups for very different purposes. Zionist historians in the early 20th century leaned heavily on the idea of continuous biological descent from ancient Israel as part of the broader case for a modern Jewish return to the Levant. Critics of that political project, including some historians writing in more recent decades, have leaned just as heavily on the opposite argument, framing Jewish identity as a purely religious and cultural category assembled from unrelated converted populations, precisely in order to challenge the historical basis of that same claim to the land. And separately, on the political and cultural fringes, versions of the Khazar hypothesis have circulated for decades among groups seeking to detach Ashkenazi Jews specifically from any claimed Middle Eastern origin altogether. None of this political weight makes the underlying scientific question any less genuinely interesting. But it does help explain why the question of Jewish genetic ancestry has been examined, re-examined, and scrutinized by population geneticists with a level of rigor and repetition rarely applied to the ancestry of any other single population in the world. The most direct and comprehensive test of these questions arrived in 2010, when a research team led by Doron Bahar published a landmark genome-wide study in the journal Nature, examining close to 500,000 genetic markers across newly collected samples from 14 separate Jewish diaspora communities, compared against 69 non-Jewish reference populations spanning Europe, the Middle East, the Caucasus, and North Africa. In the very same year, a separate and independently conducted study led by Gil Atzman examined 7 major Jewish populations, including Ashkenazi, Iranian, Iraqi, Syrian, Turkish, Italian, and Greek Jewish communities, using an entirely different sampling approach and genotyping method. Remarkably, these 2 independent research efforts, along with a third contemporaneous study led by Nama Koppelman, arrived at strikingly similar conclusions. Across all 3 studies, Jewish populations from every major diaspora community consistently clustered together into a distinct, genetically coherent grouping when compared against the broader backdrop of hundreds of world populations, despite the enormous geographic distances and centuries of separation involved. Ashkenazi, Sephardic, Mizrahi, and North African Jewish samples each formed their own genetically distinguishable subclusters, reflecting real and measurable differences accumulated through centuries of separate history. And yet, all of these subclusters consistently nested together within a single larger Jewish genetic cluster, positioned in the genomic landscape closer to Middle Eastern and Southern European non-Jewish populations than to more distant world populations, and most strikingly of all, overlapping most closely of any non-Jewish reference groups with 2 specific populations, the Druze and the Cypriots, both of whom are themselves believed to retain deep and relatively undiluted ancient Levantine ancestry. This finding directly contradicted the strongest version of the argument that Jewish communities share no meaningful biological connection to one another or to an ancient Levantine source population. If Jewish communities across the world were simply unrelated local populations who happened to share a religion, researchers reasoned, there would be no obvious reason for genome-wide data to consistently pull Yemenite, Moroccan, Iraqi, Italian, and Eastern European Jewish samples into a shared genetic cluster distinct from their respective host populations and positioned close to the ancient Levant. The fact that this pattern emerged independently in 3 separate studies using different samples, different populations, and different statistical methods gave researchers considerable confidence that the underlying signal was real, rather than a statistical artifact of any single study's specific design. The Khazar hypothesis, the more specific claim that Ashkenazi Jews in particular descend primarily from a Turkic-speaking Caucasus steppe population rather than from the Middle East, received its own dedicated and even more rigorous test in a follow-up 2013 study published in the journal Human Biology, again led by Doron Bahar and colleagues. This study specifically assembled the largest genetic dataset ever compiled for this exact question, incorporating newly collected samples from populations across the historical Khazar region north of the Caucasus, alongside the existing Ashkenazi and broader Jewish and non-Jewish reference data, precisely so researchers could directly compare Ashkenazi genetic ancestry against the specific geographic source the Khazar hypothesis proposed. The results were unambiguous. Ashkenazi Jewish samples showed their strongest genetic affinity overwhelmingly with other Jewish populations, particularly Sephardic and North African Jewish communities, and among non-Jewish populations, with groups from Southern Europe and the Middle East. No elevated genetic similarity to populations from the Caucasus region, including populations specifically chosen to represent the historical Khazar homeland as closely as modern genetics allows, was detected. The study's authors stated their conclusion plainly, finding no evidence supporting a Khazar origin for the Ashkenazi population, and instead reaffirming that Ashkenazi ancestry derives from a combination of Middle Eastern and European sources, consistent with the historical record of migration out of the Levant, followed by centuries of settlement and some degree of intermarriage within Europe. This doesn't mean the Khazar Khaganate's well-documented historical conversion to Judaism was fabricated. Historical and archaeological evidence for a Judaized Khazar ruling elite in the 8th and 9th centuries is reasonably well established. What the genetic evidence rules out is the specific claim that this Khazar population went on to become the primary ancestral source of the much larger Ashkenazi Jewish population that would later flourish across Poland, Germany, and Eastern Europe. Whatever happened to Khazar communities following the collapse of their empire in the 10th century, they did not leave behind a detectable genetic footprint at the core of the Ashkenazi gene pool. A separate but closely related line of genetic research, focused specifically on patterns of shared DNA segments within the Ashkenazi Jewish population, rather than comparisons against outside groups, added further weight to the picture of a population descended from a real historical founding group, rather than a loose assembly of unrelated converts. Studies measuring identity by descent, the specific stretches of genome that any two individuals share because they inherited them from a common ancestor, found that Ashkenazi Jews share unusually long and unusually frequent identical DNA segments with one another compared to most other populations of similar size, a pattern population geneticists specifically associate with a genetic bottleneck, meaning the population descends from a relatively small number of founding individuals, whose numbers later expanded dramatically. Estimates derived from this kind of analysis place the effective founding population of the Ashkenazi community at only several hundred individuals, with the community's numbers expanding from that narrow founding base to several million people over the centuries that followed. This same founder effect is the reason Ashkenazi Jews, despite representing a large and worldwide population today, show elevated rates of a specific set of inherited genetic conditions, a medically important and thoroughly documented pattern that would be very difficult to explain if the Ashkenazi population had instead formed primarily through the continuous, gradual absorption of large numbers of unrelated local converts across many centuries, since a population built that way would be expected to show far greater underlying genetic diversity rather than the signature of a narrow shared bottleneck. Having tested and substantially undermined the claim of zero shared ancestry, researchers turn to an equally important question on the opposite side of the argument. If Jewish diaspora communities really do share a genuine ancient connection, exactly how much of that original ancestry remains, and how much has been diluted or reshaped by many centuries of life among different host populations? Here, the genetic picture that emerged was considerably more layered than the simplest version of the traditional continuous bloodline narrative would suggest, and it is on this specific point that one particular genetic signal stood out as more consistently, precisely, and remarkably preserved than almost any other marker researchers examined. That signal involves the Kohanim, the hereditary Jewish priestly line traditionally understood to descend patrilineally from a single ancestor, the biblical figure of Aaron, with priestly status passed down from father to son across more than 3,000 years, entirely independent of which broader Jewish community a given family happened to belong to. Because priestly status in Jewish tradition is inherited exclusively through the paternal line, this specific lineage offered geneticists an unusually clean natural experiment, a single, clearly defined subgroup within the Jewish population, whose members, according to tradition, should all share direct patrilineal descent from one common ancestor, regardless of whether their families ended up in Poland, Yemen, Morocco, or anywhere else. When researchers examined Y chromosome data from self-identified Kohanim across multiple entirely separate and historically isolated Jewish communities, including Ashkenazi and Sephardic populations that had experienced essentially zero contact with one another for the better part of 1,000 years, they found something extraordinary. A specific and a relatively rare set of Y chromosome markers concentrated within haplogroups J1 and J2, both lineages associated with the ancient Near East, appeared at dramatically elevated frequency among Kohanim compared to the general Jewish population, and appeared at strikingly similar frequency across Kohanim sampled from communities with no shared history for many centuries. This pattern, now widely referred to as the Cohen modal haplotype, was first identified in the late 1990s and has since been refined through more detailed sequencing, with subsequent research estimating that the most recent common paternal ancestor of the dominant Cohen lineage lived somewhere in the range of 3,000 years ago, remarkably consistent with the traditional dating of the biblical priesthood itself. The original discovery of this pattern, published in 1997 by a research team led by Karl Skorecki, tested Y chromosome markers among Kohanim from both Ashkenazi communities in Europe and Sephardic communities in the Middle East and North Africa, two branches of the Jewish diaspora that had been almost completely isolated from one another since at least the Roman period, making the discovery of a shared and statistically distinctive paternal signature across both groups especially difficult to explain through coincidence or through parallel, unrelated conversion events. A later and more detailed follow-up study led by Michael Hammer in 2009, using more refined sequencing of the Y chromosome, estimated that the most recent common ancestor of the dominant Cohen paternal lineage lived approximately 3,190 years ago, placing the origin of this shared lineage squarely within the broad window traditionally associated with the founding of the ancient Israelite priesthood, a level of chronological agreement between an oral tradition thousands of years old and an independently calculated molecular date that few other traditions anywhere in human history have been able to claim. This is precisely the kind of evidence that a purely religious, conversion-based origin for Jewish communities would not be expected to produce. Random conversion of unrelated local populations into Judaism across a dozen different countries in many centuries should not, by chance, produce a shared and statistically distinctive paternal genetic signature concentrated specifically within a hereditary priestly subgroup and dated to a period matching the traditional founding era of that priesthood. The Cohen modal haplotype findings represent one of the most specific, most independently replicated, and most genuinely startling confirmations to emerge from the entire body of Jewish population genetics, precisely the ancient connection that stood out most clearly once researchers began comparing DNA across communities that history had kept apart for 1,000 years or more. The broader picture beyond the priestly lineage specifically turned out to be more genuinely mixed and considerably more historically interesting for exactly that reason. Autosomal DNA, which is inherited from both parents and offers the most complete picture of a population's overall ancestry, confirmed that every major Jewish diaspora community carries a substantial and clearly identifiable Middle Eastern genetic core, but also revealed that the proportion of local host population admixture layered on top of that core varies considerably from community to community, reflecting real and specific differences in each population's individual history. Ashkenazi Jews, whose ancestors settled across Central and Eastern Europe beginning roughly 1,000 years ago, show the most substantial evidence of European genetic contribution among the major Jewish groups, with multiple independent studies estimating that somewhere between roughly 40 and 60% of the Ashkenazi genome traces to European sources rather than the ancient Middle Eastern founding population, a proportion that is considerably higher on the maternal side than the paternal side. A detailed 2013 study of Ashkenazi mitochondrial DNA, led by Marta Costa, found that approximately 40% of Ashkenazi maternal lineages trace back to just 4 specific founder mitochondrial haplogroups of likely European origin, a finding interpreted as strong evidence that a meaningful number of European women joined and married into Jewish communities across the medieval period, converting to Judaism and becoming maternal ancestors of a substantial share of the modern Ashkenazi population, even as the community's paternal lineages and its cultural and religious identity remained much more consistently rooted in the Middle Eastern founding population. Sephardic Jews, whose ancestors settled across the Iberian Peninsula before their expulsion in 1492 and subsequent dispersal across North Africa, Southern Europe, and the Ottoman Empire, show a genetic profile that researchers have consistently described as intermediate between Ashkenazi Jews and non-Jewish Middle Eastern populations, carrying a somewhat smaller proportion of European admixture than Ashkenazi communities, and a correspondingly larger share of retained Middle Eastern ancestry. Detailed Y chromosome research on Moroccan Jewish communities specifically, drawing on genealogically verified samples through dedicated genetic genealogy projects, found that roughly 70 to 80% of paternal founder lineages traced to haplogroups common across the Middle East, with a further meaningful share, around 11%, tracing specifically to the Iberian Peninsula, reflecting the well-documented historical exchange between Iberian and North African Jewish communities across many centuries, while genuinely local North African paternal ancestry accounted for only a small minority of lineages, directly contradicting an older, and now largely abandoned theory that North African Jewish communities form primarily through mass conversion of indigenous Berber populations. Mizrahi Jewish communities, those whose ancestors remained in the broader Middle East itself, in countries including Iraq, Iran, and Syria, unsurprisingly showed the closest genetic affinity of any Jewish subgroup to non-Jewish Middle Eastern and Caucasus populations, and the least evidence of European admixture, consistent with a population that never left the general region their ancestors originated in and experienced a very different demographic history from the communities that eventually settled in Europe. A handful of more geographically distant Jewish communities produced some of the most genuinely fascinating and complicated results in the entire body of research. The Beta Israel community of Ethiopia and the Bene Israel and Koshin Jewish communities of India each maintain strong religious and cultural traditions connecting them to ancient Israel. Yet genome-wide analysis has consistently found that these specific communities genetically resemble their surrounding local populations, Ethiopian and South Asian respectively, far more closely than they resemble the broader Middle Eastern Jewish genetic cluster identified in the Ashkenazi, Sephardic, and Mizrahi communities. though some researchers have identified limited traces of a more ancient shared signal in more detailed analyses of specific communities. This finding illustrates something important about how differently the same underlying question can resolve itself across different Jewish communities depending on their specific historical circumstances, the size of the founding population that established each community, and the degree to which each community maintained strict religious endogamy across the centuries that followed, or instead incorporated substantial numbers of local converts into a smaller founding nucleus over time. One further wrinkle in the story adds real depth to the picture, and it concerns communities that appear on the surface to fall outside the traditional Jewish diaspora entirely. Across Spain, Portugal, and large parts of Latin America, historical records document the existence of so-called converso and crypto-Jewish communities, families forcibly converted to Christianity during the Iberian expulsions and inquisitions of the late 15th and 16th centuries, some of whom are recorded as having continued practicing elements of Jewish tradition in secret across many subsequent generations, while publicly identifying as Catholic. Modern genetic studies examining Y chromosome data from present-day populations in Spain, Portugal, and parts of Latin America, with documented converso ancestry or regional historical association with forced conversion, have found a measurable elevated frequency of the same Middle Eastern paternal haplogroups characteristic of the broader Sephardic Jewish population, including lineages within the same broad Cohen-associated haplogroups discussed earlier, at levels higher than would be expected from general regional background alone. This finding offers an unusually direct genetic window into a specific and historically well-documented episode of forced religious conversion, showing that a meaningful paternal genetic signal from an originally Jewish population can persist for many centuries, even after a community's religious and cultural identity has been formally and often violently erased. A finding that adds a genuinely poignant dimension to the broader question of how ancestry and identity relate to one another across long stretches of difficult history. The internal genetic structure revealed across all of these communities together also helps explain why the debate over Jewish ancestry proved so resistant to resolution through historical argument alone, long before genetic tools became available. Because the honest answer was never going to be a simple yes or no to either extreme claim. A population descending entirely and exclusively from local conversion across a dozen unrelated regions would leave a very specific, very identifiable genetic signature, essentially indistinguishable from each surrounding host population with no shared clustering at all. A population descending in complete isolation from a single, unchanged ancient bloodline with literally zero admixture across 2,000 years spent as scattered minorities inside dozens of much larger host societies would leave an equally specific and equally identifiable signature, one showing no meaningful genetic distance from ancient Levantine populations and no meaningful genetic similarity to any local host population whatsoever. Neither of those two clean, mutually exclusive signatures is what the data actually show. What emerges instead, consistently across 3 independent major studies and dozens of smaller follow-up analyses conducted over more than 15 years, is a population structure that sits precisely between those two extremes, exactly the pattern that a genuinely dispersed people maintaining strong internal marriage practices while still absorbing a real and measurable degree of local admixture at different rates in different times and places would be expected to produce. Taken together, this accumulated body of research points toward a conclusion that is considerably more precise and in several genuine respects more remarkable than either side of the century-long argument over Jewish ancestry had fully anticipated. The claim of a single, unbroken, essentially unchanged bloodline running in isolation from ancient Israel through every subsequent generation in every diaspora community does not hold up against the genetic evidence, which clearly shows real, substantial, and historically specific admixture with local host populations occurring at meaningfully different rates across different Jewish communities, most dramatically on the maternal side among Ashkenazi Jews, and most distinctly among the more geographically distant Ethiopian and Indian Jewish communities. But the opposing claim that Jewish communities share no deeper biological connection at all beyond religious practice fares considerably worse under the same scrutiny, decisively contradicted by the consistent clustering of Ashkenazi, Sephardic, Mizrahi, and North African Jewish populations into a shared genetic grouping positioned close to ancient Levantine reference populations, by the specific and repeated refutation of the Khazar hypothesis for Ashkenazi origins, and above all by the extraordinary and independently replicated persistence of the Cohen modal haplotype among Jewish priestly lineages separated by continents and by well over 1,000 years of independent history. None of this diminishes the cultural, religious, and personal significance that shared ancestry, and equally, the story of dispersal, survival, and local integration continue to hold for Jewish communities and individuals today. Origin narratives, whether emphasizing an unbroken bloodline or emphasizing a history of local integration and conversion, serve purposes that extend well beyond strict genetic accuracy, offering communities a sense of identity, continuity, and belonging that remains meaningful regardless of exactly what a Y chromosome or mitochondrial analysis ultimately confirms. What the genetic evidence does offer is a far more precise, evidence-based map of exactly which threads of ancestry actually connect these scattered communities to one another and to an ancient Levantine source, and which threads instead reflect the very real and very human history of 2,000 years spent living, marrying, and in some documented cases, converting, alongside dramatically different host populations across 4 different continents. Not a single sealed and static bloodline preserved in isolation for 2 millennia, and not a scattered collection of entirely unrelated communities joined only by shared religious texts, but a genuinely dispersed people whose modern genetic diversity reflects the accumulated weight of 2,000 years of separate history, layered on top of a shared and now scientifically confirmed ancient Levantine core that never fully disappeared, no matter how far its carriers eventually traveled from the region where that story first began. The Cohen modal haplotype findings in particular carry a significance that extends well beyond the specific question of priestly descent. They demonstrate, with a level of precision that no amount of historical or religious argument alone could ever have supplied, that at least one very specific line of oral tradition maintained independently across Jewish communities that had no contact with one another for the better part of a millennium corresponds to a real, measurable, and datable genetic signal. That is the real genetic story connecting Jewish communities across the world today, not a single unchanged bloodline sealed off from every population it encountered, and not a set of unrelated peoples joined by nothing more than shared scripture, but a genuinely scattered people, reshaped in different ways by different corners of the world they came to call home, who nonetheless carried something real and specifically traceable back to a shared point of origin in the ancient Levant, a connection precise enough that modern genetics has finally been able to measure exactly where and exactly how strongly it survived, buried beneath 2,000 years of separate history, yet never entirely erased by any single century of it, discovered at last not through the argument of historians or the assertion of tradition alone, but through the quiet, patient comparison of one genome against another, community by community, until the pattern connecting all of them finally became impossible to ignore.